Dear students, today we will discuss electron transport chain in cellular respiration. So the glucose metabolism started with the glycolysis. In the glycolysis, the end product is pyruvate. The pyruvate is converted to acetyl-CoA and acetyl-CoA enters the Krebs cycle. Now, whatever reducing powers that were produced during the glycolysis and Krebs cycle in the form of NADH plus H and FADH2, they finally enter electron transport chain and transfer their electron to the series of electron carrier. So let's see what is electron transport chain, which is also known as mitochondrial respiratory chain, as it occurs in the inner membrane of mitochondria. So, electron transport chain is the series of complex that consist of coenzyme and protein located in the inner membrane of mitochondria that transfer the electrons from electron donor to electron acceptor via redox reaction. The electron transfer is coupled with the transfer of protons across the membrane. The transfer of proton produces an electrochemical gradient for hydrogen ions. This gradient is used to generate ATP from ADP plus Pi. Pi means inorganic phosphate. Electron transport chain is the only part of glucose metabolism that uses atmospheric oxygen, or we can say free oxygen, as the final electron acceptor. So electron transport chain, basically three different steps, or you can say three different things are involved. One is generating the proton motive force. Second is ATP synthesis via chemiosmosis hypothesis and reduction of oxygen. So we will see what all components are there of electron transport chain. So basically it consists of electron acceptors and respiratory coenzyme complexes. So first we will discuss the electron acceptor. The electron acceptors are NAD and FAD. Basically they are the primary electron acceptor and they pick up the electron from the mitochondrial matrix that are released from the substrate. After accepting the electrons and protons, they are reduced to NADH and FADH2 as we have seen in the Krebs cycle. They transfer their electrons and proton in electron transport chain. So NADH plus H and FADH2, they are reduced form of coenzyme, and they are produced during the Krebs cycle. They are present in the mitochondrial matrix, but they finally transfer their electron finally in the electron transport chain. So when they transfer the electron, what will happen? NADH plus H, okay, it gives out two electron and two protons and get converted into NAD plus. Similarly, FADH2, it gives or you can say donate two hydrogen and uh, two hydrogen ion, and or we can say proton and electrons, and produces FAD. And finally these electrons and proton are accepted by oxygen. Oxygen is the finally acceptor, you can say, and then once the oxygen accept these electron and proton, it get converted to water. So this is how the water is formed during the cellular respiration. Now let's discuss respiratory coenzyme complex. There are total 4 complex. We'll discuss one by one. Complex 1, which is known as NADH dehydrogenase complex. Complex 2, which is known as succinate dehydrogenase. It is also known as succinate Q reductase. Complex 3, which is known as cytochrome B to C1 or coenzyme Q to HZ or cytochrome reductase complex, and complex 4, which is known as cytochrome A to A3 complex. Now if you look at the location of respiratory coenzyme complex, see, this is the mitochondria. You can say this is the outer membrane and then inner membrane of mitochondria and mitochondrial matrix, and the space between the inner and outer membrane is called intermembrane space. So if you look at the location of these complexes, complex 1, 2, 3, 4, and they are present in several copies. Okay, so this is how they are located, with the as a membrane bound proteins, especially to the inner membrane. Okay, now we will discuss uh, what these complexes consist of. So if you look at complex 1, it consists of FMN, that is flavin mononucleotide, and FES, that is iron sulfur proteins. It catalyzes redox of NADH plus H, and the electrons are transferred to complex 2, but the protons are pumped to the intermediate space. We have already seen that two electron and one proton is transferred to the next, you can say, complex 2 or next electron acceptor, whereas one proton is pumped in the intermembrane space. Okay, if you look at complex 2, consists of ubiquinone, which is um, written as Q, and it accepts the electron and proton from complex 1. Now complex 2, one first part is that it can accept the proton and electron from complex 1, and second, it directly accept the electron and proton in the form of FADH2 directly. They give their electron, FADH2 directly give their electron to complex 2. Okay, so complex 2 do not pump the proton to the intermediate space. So this is the only complex which is not pumping any proton to the intermediate complex. Okay, now if you look at complex 3, it consists of cytochrome B1, cytochrome C, and iron cellular protein cytochrome C accept the electron and proton from Q, that is complex 2. The proton are pumped to intermediate space. Now, if you look at complex 4, it consists of cytochrome C1, A, and A3, and also heme and copper ions. Cytochrome hold on oxygen molecule very tightly between the iron and copper ions until the oxygen is completely reduced. Oxygen pick up proton H plus, that is proton, from surrounding and make the water. The rest of the proton move to intermediate space. Now, if you look at here, see, this is the inner membrane. Suppose this is the inner membrane. You can look at this diagram. Here is the inner membrane. So here I am showing you, this is suppose the inner membrane, and these are the complexes, 4 complexes which are located. So you can say complex 1. Okay, so it redox NADH plus H, and NADH plus H is converted to NAD. Okay, this is converted to NAD. Okay, and NAD is again used up in the Krebs cycle. It is present in the mitochondrial matrix. Okay, so when this is doing this, the electron and protons are released. 2 electron and 1 proton is transferred to complex 2, whereas 1 proton will move to the intermembrane space. Okay, now the complex 2, so it receives the electron from complex 1, as well as it also directly receives the electron from FADH2. So FADH2, once it donate 2 electron and proton, uh, and it get itself converted into FAD. Okay, so now these electrons are transferred to complex 3. Complex 3, there are several proteins, carrier proteins, and finally, while transferring the electrons and proton, they pump the proton in the inner membrane space. Similarly, complex 4 also do the same thing. So here, since copper and iron are there, they can bind the oxygen. So oxygen is present here, and which is the final electron acceptor. So finally, the electron and protons are accepted by oxygen, and oxygen itself get reduced to water. Now this reaction is catalyzed by the enzyme cytochrome C oxidase. Okay, and again while doing this, whatever proton is there, and that extra proton is released in the intermembrane space. So if you see in the intermembrane space, because there are a lot of hydrogen ions which are accumulated over here, or you can say protons which are accumulated here, because hydrogen ions are protons. Okay, so this increases the concentration of hydrogen ions, and we can say high concentration of hydrogen ion means lower is the pH of this particular intermembrane space as compared to matrix where the concentration of H plus ion is lower. Here one thing to note, that cytochrome C oxidase is the very important enzyme, and cyanide, you must have heard of a chemical which is, so oxygen is the final acceptor of electron transport chain. It accepts the proton and electron and reduce to water. So this is how the water is produced during the cellular respiration. Now this reaction is catalyzed by the enzyme cytochrome C oxidase. It is You must know that cytochrome C oxidase is the enzyme, very important enzyme, and the cyanide, which is a kind of chemical compound, it inhibits the activity of cytochrome C oxidase. So immediately if there is cyanide in any cell, then it inhibit this activity and stop the ATP synthesis, because this will cause alteration in the pH of intermembrane space and matrix, and the enzyme ATP synthetase will not be able to catalyze the synthesis of ATP. So the next is we will discuss about the ATP synthetase enzyme. Now the concentration of proton increases in the intermembrane space as compared to the matrix, and this uneven distribution of protons across the membrane establishes both concentration and electrical gradient. So that's why it is known as electrochemical gradient. As a result, the H plus ions are diffused back to matrix, but this pumping back of H plus ions occur by ATP synthesis. Now, ATP synthesis is also known as molecular motor of living cell, as it acts as a generator which is turned by the force of H plus diffusing through it. Okay, that is by the protons, or we can say hydrogen ions. Now, ATP synthetase is the integral membrane protein that catalyzes the formation of ATP. As what it do, the ADP plus inorganic phosphate forms ATP in presence of enzyme ATP synthetase by using the proton motive force. Now, what is proton motive force? Proton gradient, or we can say the gradient by H plus ion generated by the oxidation of NADH and FADH2 is called proton motive force. Basically, it is the energy of proton concentration gradient. The proton motive force powers the ATP synthesis. Now, if you look at the structure of ATP, then it has got two subunit. One is F1 and F2. F1 is knob, forming knob. F2 forms the stalk, that is the base. So F1 subunit is projected into the matrix region. It has various subunits, 3 alpha, 3 beta, gamma and delta, and the beta subunit has catalytic activity. F0 subunit is embedded in the membrane and it acts as a channel for transport of H plus ion. Now, if you look at the structure, it will be clear. We have seen that in the intermembrane space, there are H plus ions which are accumulated, or we can say protons are accumulated. So, once there are proton accumulated in the intermembrane space, they create a force which is known as proton motive force, and because of this force they are pumped back, or you can say diffused back to the matrix. Okay, they went to matrix. From the matrix, they went to intermediate space. How? They went through the electron transport chain. Now, once their concentration rises in the intermembrane space, we say this is creating a gradient. Okay, which is also known as the electrochemical gradient. Now they diffuse back to the matrix, and this diffusion is across the membrane is now via the ATP synthetase enzyme. It is the membrane bound enzyme. Two subunits are there. F0, which is a stalk, is present in the inner membrane. F1 is projecting toward the matrix and it has various subunit, and beta subunit has the catalytic activity, so it converts the ADP to ATP by addition of inorganic phosphate. Okay, so this is how the ATP is synthesized. Now, transport of electron from one molecule of NADH plus H over ETC help in the transport of three pairs of protons from outer mitochondrial chamber to the matrix, and this generate 3 ATP molecule. Whereas transport of electron from one molecule of FADH2 help in the transport of two pairs of protons and generate 2 ATP. So per molecule of NADH plus H generate 3 ATP, whereas per molecule of FADH2 generate 2 ATP when they transfer their electron through the electron transport chain. Now, Peter Michel in 1978 awarded Nobel Prize for chemoosmotic coupling hypothesis. Now, according to this hypothesis, ATP synthesis is coupled with the proton gradient generated by high concentration of H plus ions or protons on F0 site. That is the F0 side is the stock, okay, as compared to F1. F1 is the matrix side. Now, what is oxidative phosphorylation? The phosphorylation means addition of phosphorus of ADP with Pi, that is inorganic phosphate, during the aerobic respiration to form ATP in mitochondria is termed as oxidative phosphorylation, and the energy for the synthesis of ATP comes from the oxidation reduction reaction that occur during the transport of electron from one complex to next of the electron transport chain. Therefore, together, the electron transport chain and chemoosmosis make up oxidative phosphorylation. The process of oxidative phosphorylation is only get completed in presence of oxygen because oxygen is the final hydrogen ion acceptor. Now, just how the phosphorylation differs from the oxidative phosphorylation. Phosphorylation refers to the addition of phosphate to substrate or ATP formation from ADP plus Pi at any step in the metabolic reaction. For example, we have substrate level phosphorylation, which occurs in glycolysis, which occurs in Krebs cycle also. There are reaction which produces ATP. We have seen them. Similarly, we have another term called photophosphorylation, which occurs in presence of light during the photosynthesis. So we'll know more about this during the photosynthesis. So this is all about the electron transport chain. Next, we will be calculating the total number of ATP that are produced during all these reactions. Thank you.